Bueno, pues muy buenas tardes a todos y muy bienvenidos al Instituto Cervantes. Para mí es un placer y también un honor volver de nuevo con el ciclo de influencers españoles en el ámbito de la ciencia, que está organizado por la Red de Científicos Españoles en Bélgica, CEBE, y apoyado por la Consejería Cultural y Científica de la Embajada de España en Bélgica, la Fundación Ramón Areces y, por supuesto, el Instituto Cervantes. Esta es ya nada más y nada menos, yo lo he tenido que leer 2 veces, la 7ª edición. Yo creo que eso habla de la tenacidad de nuestros científicos, de que han visto claramente que la difusión es necesaria, que la ciencia tiene que llegar a pie de la calle para que comprendamos que es, yo siempre lo comentaba ahora con una compañera, el arte de procurar que la gente pueda vivir más y mejor, y que además es necesario que conozcan todo ese trabajo arduo que se hace desde vuestros distintos ámbitos y que resulta absolutamente necesario. Hoy nos vamos a adentrar en un tema que ha levantado bastante expectación y es ese fascinante campo que explora las posibilidades de la vida más allá de la Tierra. Para ello contaremos con el investigador español Carlos Briones. Buenas tardes, muchas gracias por estar aquí. Él pertenece al Consejo Superior de Investigaciones Científicas, trabaja en el Departamento de Evolución Molecular del Centro de Astrobiología, asociado al programa del mismo nombre de la NASA, y la investigadora belga y profesora titular del Departamento de Geología de la Universidad de Lieja, Emmanuelle Jabot, bienvenue, soy bienvenue, que preside el grupo de investigación Early Life Traces and Evolution Astrobiology. Y ya, sin más dilación, voy a pasar la palabra al doctor Jorge Barrasa, del Departamento de Ingeniería Mecánica de la CAU Leuven, y como bien sabéis muchos de los que estáis aquí, responsable de comunicación de la Red de Científicos de Españoles en Bélgica. Muchas gracias.
I'm going to switch to English already, so whoever is not a Spanish speaker can relax now. So first I will start apologizing for being here because I'm not supposed to be here. The president of CEBE is very sorry to not be able to come today, so I'm going to try to do my best to present what is CEBE. So I wanted to give a little introduction of what CEBE is. CEBE is an association of Spanish scientists in Belgium. We're a non-profit organization, and we basically aim to bring science closer to the public, among other things. We do a few activities, science communication events like the one that you're going to see today. We also have professional development, and actually, if you're interested, we have in November a coming event where we will give an introduction to European institutions and oriented to scientific profiles to inform them of how to access them. We also do scientific diplomacy to, again, connect Spanish scientists in Belgium with institutions in Belgium and also in Spain, for those who are planning to go back. And then we also have the CEBE community. Basically, it's the network of all the scientists who are in similar situations can be very helpful, and we actually have something that we call CEBEZAS, which is basically gathering to have beer in different cities in Brussels, as you see here. So if you would become a member, here we have the next dates of our CEBEZAS, so you'll be very welcome to join us there. And yeah, I mean, I don't want to extend this too much because we're here to listen to our speakers here. I will start introducing, he has been already introduced in Spanish, but I will do it in English. Carlos Briones is a scientific researcher in CSIC, in the Department of Molecular Evolution and at the Center of Astrobiology in Spain. And his group has a broad background in the field of astrobiology, and they focus on molecular evolution, collaborating on interdisciplinary projects aimed at analyzing microbial biodiversity in extreme environments and detecting relevant prebiotic molecules in the interstellar medium. And he has participated in numerous research projects, and he's author of multiple scientific articles and popular science books. And this is not in his biography, but in my opinion, he's one of the best science communicators that we have in our country, in Spain. He's also a member of executive boards, including the European Astrobiology Institute. So without further ado, Carlos, I'm very honored to ask you to come on stage, and the floor is yours.
Okay, well, I will start in Spanish a little bit, because algunos de vosotros estáis estudiando español y seguro que os gusta escuchar español con acento de Burgos, español profundo. Solo para agradecer a Ana Vázquez, a todo el staff del Instituto Cervantes, por acogernos aquí en este bello lugar, ¿no? con luz y agua alrededor, ideal para hablar sobre la vida, por cierto. Y por supuesto a CEBE, a Jorge y a todo el personal de CEBE, pues por organizar esto, por traer a lo que ellos llaman influencers, que es mucho decir, pero bueno, científicos o personas que tenemos interés por la ciencia de España, y por hacerlo además joining us with colleagues here in Belgium. So it's my pleasure to be here with Emmanuel Javot, my friend, colleague, and one of the persons in science which I admire most, because she's one of the leaders of finding traces of life, of very ancient life, and perhaps life elsewhere. So thank you again to all of you for being here, and I will switch definitely to English. And we were together in a conference, some months ago, and we took that picture. So, okay, I will try to use this. Okay.
No, the left one. Left one.
Ah, left one.
Yeah, but now you're
sure?
in the... Yeah, but now you're in the... Just put up this. Oh.
I usually use one pointer, you know.
Yeah, me too.
Yep. Okay. So, I have to tell you next.
No, try to click
okay.
now. On the left. On the left button.
Okay, so I will try. I will do my best. We're going to talk about astrobiology. What is astrobiology? Astrobiology is a new science, or a sum of sciences. You see here kind of Newton wheel with mathematics, physics, chemistry, engineering, biology, geology, and philosophy surrounding everything. If you mixed everything a little bit, you get interdisciplinarity among different disciplines, and if you mix more and more and more, you get astrobiology. You know? So, astrobiology is a bit of everything. In my center, for instance, in Spain, in Center for Astrobiology, we work together, colleagues from different disciplines, and it's very easy to have in a round table a physicist, a geologist, and microbiologist, and even a philosopher, for instance. And this is very, very nice. Why? Because we're trying to understand the basic questions of the universe. We're trying to get answers to big questions such as what is life, how life originated, how life evolves. Is it possible to find life elsewhere? You need everything there. Every brain is welcome. So this is a general picture of astrobiology drawn by colleagues at NASA. And in particular, what refers to the origin of life, which is one field I have been working in for years, and also Emmanuel and other colleagues. The questions are very, very relevant. When, how, was it the outcome of chance or necessity? Okay, this is a basic question also in the field of philosophy, where the origin was terrestrial or not, once or several times. So, very, very difficult questions. We can approach this problem from the past using bottom-up approaches. This is usually performed by astrochemistry, geochemistry, prebiotic chemistry, or in recent terms by synthetic chemistry, trying to build bugs in the lab, you know. And also from the present to the past, the so-called top-down approach, using, for instance, paleontology, and Emmanuel is an expert in micropaleontology, microbiology, phylogenetics, or minimal cell models. I could talk about the origin of life for weeks, but I think that it's better to show you a short video, a 5-minute video. It's a cartoon video, so it's nice to see, I think. We have prepared at CSIC in Spain that I think recovers the main aspects related to the research on the origin of life. So, please, this is the video, and if this works. Great.
The origin of life. The mystery of the origin of life has been the subject of many different theories, which are varied. Cosmic dragons, colossal turtles, clay beings. But of course, rational thought had something to say. Aristotle put forward the hypothesis of the spontaneous generation of life, which would be refuted by Pasteur in the mid-19th century. But the first scientific ideas came from Charles Darwin. Life may have been initially breathed in a short number of forms, or in a single one. Later on, Darwin ventured to write in a private letter an intuition that his scientific rigor would not allow him to publish officially. In a warm little pond, inert matter with the help of energy sources would organize itself into living matter, capable of undergoing more complex changes. Decades later, other researchers followed in his seminal work, proposing the first models for the origin of life. Experiments came later. Miller inaugurated the field of prebiotic chemistry, mixing 4 gases in a glass vessel formed by 2 flasks. Applying continuous electrical sparks, many of the amino acids that make up proteins were produced, together with other small molecules that could evolve into LUCA. LUCA? Who is LUCA? Last, universal, common ancestor. LUCA would be the last common ancestor of all living beings on this planet. What would this form of life look like, considering that it already contained everything that all living entities since then also possess? No, LUCA probably looked a little more like this. From LUCA, 2 large branches of microorganisms without a cell nucleus, the bacteria and the archaea, were separated. Much later, a 3rd group originated, the eukaryotes, which includes microorganisms with a nucleus and also multicellular beings, plants, fungi, and animals. This tree of life is not linear because organisms exchange genes, sometimes using viruses as carriers. So in reality, the image that best reflects biological evolution would be more like a complex vine. How could we, in this tangle, find LUCA and understand how it came into being? Let's change perspective. Let's try to get LUCA from the past. About 4.5 billion years ago, the Earth was formed. About 100 million years later, it cooled enough to have a solid crust and large oceans. We assume that LUCA lived about 3.6 billion years ago. On the way from primitive Earth to LUCA, the first stage was the formation of small organic molecules in different environments, until a more or less complex repertoire of compounds was generated in a solution that we know as the primitive soup. More than 300 organic molecules have been identified in space so far. Some of the soup's ingredients probably arrived on Earth via meteorites or cometary nuclei. This led to the formation of increasingly large and complex molecules capable of performing different biochemical functions. Among them, the RNA world model proposes that this biopolymer appeared before proteins and DNA. Prebiotic systems chemistry offers a more complex and realistic view of the origin of life. It posits that 3 systems had to be combined in the first living beings: replication, metabolism, and compartment. This combination would give rise to the first living beings, chemical systems capable of reproducing and evolving. Some of them would eventually give rise to LUCA, a species with a flow of genetic information following the path DNA, RNA, protein. LUCA could be either autotrophic, using chemical energy present in its environment, or heterotrophic, feeding on available organic compounds. Each theory leads us to different scenarios for the origin of life. from underwater hydrothermal vents to the warm little ponds suggested by Darwin. But are we the result of chance or necessity? If life was the result of many coincidences, perhaps Earth is somehow a singularity, the only living planet. But if life is a necessary consequence of increasing chemical complexity, we're probably surrounded by other life in the universe. In this field, we do interdisciplinary research, combining physics, astrochemistry, geology, prebiotic systems chemistry, biochemistry, microbiology, astrobiology, and philosophy. Scientific organizations all around the world, such as the CSIC in our country, work in coordination to get closer to the mystery of life and its origins.
Okay? Yeah? Thank you. It was a pleasure, in fact, to prepare this short video. Okay, so life originated in the bulb. No? Again, no.
Now it's gone down.
Again and again until you fix all your ideas. So, if we combine some disciplines, for instance, astrochemistry, prebiotic chemistry, or the origins of life, and biology, at the end, we can have an idea about this process in our planet, and perhaps in other planets or other bodies of a solar system or beyond. And the key issue here is that once life is originated, living beings produce molecules that cannot be produced by chemistry itself. These molecules are called biomarkers. Okay, for instance, some vitamins, for instance, some aggregates of amino acids called proteins or nucleic acids and so on. Only living beings are able to produce them. So if you find elsewhere one biomarker, you can say, okay, perhaps we have found life. Okay, so this is a key issue, and I will show you some examples of which kind of molecules have been found in different places, telling us that perhaps chemistry is very complex, but not complex enough to have originated life. Okay? If we focus on the interstellar medium, we can find different molecules. You see here a small repertoire of different molecules that have been found. Here you have a list of about 120 different molecules that you can find if you focus your telescopes to some clouds in the universe. Okay. The number of carbon atoms here is in general from 1 to 6, with some exemptions, because you have also 8, 9, 10 atoms in total, and some of them have more carbon atoms. And you have 60, 70, so fullerenes, buckyballs, formed only by carbon. Okay? But it's very interesting to find these molecules elsewhere. But it's also interesting because once you find, for instance, urea, okay, in a cloud, it seems that these molecules are not biomarkers because the chemistry has been able to produce these molecules. So if you find the same repertoire of molecules, for instance, on Mars, you cannot say that I have found life on Mars, because chemistry can do it by itself. Okay? So this is the pros and cons of finding molecules in the universe. For instance, I was involved in the discovery of ethanolamine in the space with some colleagues, some astrochemists at Center for Astrobiology. We were able to analyze these signals and say, okay, in the center of our galaxy, there is a source of ethanolamine. The ethanolamine is the head of the lipids that form the membranes of our cells. So, okay, parts of the ingredients of early life could have been delivered by perhaps meteorites or cometary nuclei to our planet. Okay? So, ethanolamine is not a biomarker. Chemistry produced ethanolamine in the center of our galaxy. Even a molecule very related to our life is not a biomarker. So be careful if you find ethanolamine in other places. This is one of the open questions in astrobiology. Okay? For instance, you can go to asteroids. This is, well, in fact, in 2 days, we will receive, in 4 days, we will receive the samples of an asteroid, okay? The asteroid Bennu has been visited by a mission called OSIRIS-REx by NASA, but the previous one, the previous asteroid visited was Ryugu by Hayabusa 2 mission from the Japanese the Japanese space agency, okay, space agency called JAXA. And when analyzing the rocks that were taken at the surface of this asteroid once on Earth, okay, they were able to find uracil and vitamin B3. So 2 molecules which are essential for our life, for all life in our planet, and they are on an asteroid. So they are also not a biomarker, okay? We are investigating Mars in astrobiology. We don't have to talk in depth about Mars, but the only thing I want to tell you is that Mars is as you see here in the present. This is a composition of pictures taken by Viking orbiters. But when life originated here, as you saw in the video, about 3.8 billion years ago, Mars was like this. All the northern hemisphere of Mars was covered by a giant ocean. And probably life also originated on Mars. If life originated on Mars, if, a big if, as Darwin said, a big if, perhaps life is already there, in the subsurface, okay? So we have to find biomarkers on Mars, but real biomarkers. The evolution of Mars has been very different to that of the Earth. So Mars is not habitable. The surface of Mars is not habitable currently, but it was in the past, and perhaps the subsurface is fully habitable. For instance, methane has been discovered on Mars, some pulses, some short periods of production of methane. But the point is that methane is not a biomarker. So those saying, okay, if there is methane, is because there are bugs, microbes producing methane, because methane here is produced by a certain type of microorganisms. I'm not going into details. Say, okay, so if you find methane, you find life. No, because methane can be produced chemically, as you see. A reaction called serpentinization can produce methane, okay? Perhaps the answer to the question of life on Mars could be answered, perhaps, in some years, when missions coming from this Mars Sample Return project are recovered on Earth, okay? And we will be able to analyze about 60 pieces of soil and subsoil of Mars, near surface samples, and be able to apply all our technologies and say, okay, are there molecules really related to life here on those samples? Let's see. About Venus, Venus is like hell. The surface is 400 degrees Celsius degrees temperature, so it's not possible to find life here in Venus. Even very extremophilic life will die if they go to Venus. But the possibility could be the clouds at about 50 kilometers above the surface. And it was quite famous, this paper 3 years ago, claiming that they have found phosphine, phosphorus and three hydrogens, pH3, this molecule, and said, okay, as phosphine is produced on Earth by living beings, the finding of phosphine means that we have found life in the clouds of Venus. And it was not the case, because phosphine is not a biomarker. So there are chemistry, there are different chemical reactions that can produce phosphine. Okay, so we have to be very, very careful. Okay. Next example, Europa, the satellite of Jupiter. We are very interested in this kind of satellites covered by ice and with a liquid water ocean beneath the surface. In the case of Europa and of course Enceladus around Saturn, we have been able to find this kind of geysers of water vapor coming from the subsurface. And in principle, perhaps we have all the chemistry required to produce life in this kind of environments. We don't know if there is life, but the possibilities are open. And Europa and all these ocean worlds, as we call them, have the three main ingredients of life: water, chemistry, I mean carbon-based molecules, and also energy. So perhaps there is life on Europe. Okay, we have to find. In fact, European Space Agency sent this mission some months ago, JUICE, that will reach the system of satellites around Jupiter in 2031. So we have to be patient to see if there are some signs of life on Europe. And NASA is going to send Europa Clipper next year with a similar, let's say, objective. Okay. Because there are lots of water in the solar system. Water is very abundant in the universe, and in particular in our planetary system. So the preconditions to find life in different satellites or planets in our planetary system is quite high. Okay. And finally, extrasolar planets. Okay. There are more than 5,000 planets already discovered and characterized. In fact, more than 5,500, because this was prepared when the planet number 5,000 was discovered in 1.5 years ago. But the expected number of planets in the universe is 10 to the 23. Okay? One followed by 23 zeros. This number is enormous, it's astronomical in all senses. And the possibilities to find life in any of those planets is huge, okay? So we have to be careful, because we have to try to find life in places which are very, very far away from here, getting all information about the chemistry of their atmospheres in the best case, okay? There are different factors affecting habitability of a planet. We are not going into details of it, but it's a complex issue. And every year or couple of years, they prepare the list of the potentially habitable exoplanets. And potentially habitable means potentially habitable, not more than that, not inhabited. Okay. You have a very good example, very close to this country, because the TRAPPIST system was discovered by Belgian astronomers working in a couple of Belgian telescopes in Chile, called TRAPPIST. I don't know the reason why, but okay. This TRAPPIST system has at least 2 planets that are potentially habitable, okay? And at a distance at which water can be in liquid state at the surface, okay? Perhaps there is life there. Perhaps there are bugs very happy in the TRAPPIST D and E planets, but we don't know. And just to finish, a couple of weeks ago, James Webb Telescope, this wonderful telescope of NASA and also European Space Agency, found one exoplanet called K2-18b, and the atmosphere of this exoplanet contains CH4, so methane, and also CO2, okay? This means that there are organic molecules. This means that complex chemistry perhaps can be developed there, and this is very, very good news. And this is very good news that James Webb is able to analyze the atmosphere of a planet which is very, very far away. But this doesn't mean that we have found life there. Perhaps you have heard or read news telling, okay, NASA found life or evidences of life in an extrasolar planet. No, okay? So to end my part of this talk with this nice picture, 3 take-home messages, okay? First, potentially habitable worlds are not necessarily inhabited. Okay, it's easy, but we have to repeat again and again and again. We work in astrobiology because some journalists are very careful with that, but others prefer to say, why not saying, okay, inhabited, okay, because it's more sexy, okay? No. Then, we haven't found any evidence of extraterrestrial life, okay? Even in the Congress of the States or in Mexico. No such evidences. And finally, something that philosophers told us about 400 to 500 years ago. oh, sorry, backwards, please. Extraordinary claims require extraordinary evidence. Okay? We don't have to forget this, because this is a key issue in science in general, and in particular in astrobiology. I would like to thank lots of people, blah, blah, institutions, and so on, and of course, I would like to thank you for being here. This is a picture of my center in spring. Okay, a bit more sunny than this part of Europe, and you are cordially invited to visit us when you want. Okay, so thank you very much, and I give the floor to my friend Emmanuel.
Thank you, Carlos, for this very inspiring talk. Now, well, as he already said, our next speaker is Professor Emmanuelle Javot. She's a researcher and full professor in the Department of Geology in the University of Liège, and her research group, the Early Life Traces and Evolution Astronomy, is dedicated to study the early stages of life and evolution in astrobiology. She's also a member of the European Astrobiology Institute, and with an outstanding academic career. I actually invite you to check her publications in Google Scholar. It's quite impressive. She's received numerous awards and recognitions for her work in the fields of paleobiology and astrobiology. And in addition to leading national and international projects, she plays important roles in scientific organizations and groups related to astrobiology. And I would like to give Emmanuelle a special thanks to be here, because it wasn't easy for her to be here today, but she's here for us. So the floor is yours now, Emmanuelle. Thank you.
Can you hear me? Yes? Yeah, okay. So thank you very much for the invitation, and I just would like to point out that you have the sun, but we have the beers and cervezas and chocolates and the good mood. So it's a nice idea to mix both of countries, the good sides of them. So there will be some overlaps between my presentation and Carlos' presentation, but perhaps it's good to see things different ways, the same thing different ways, and of course there will be some differences. I'll push on the left side. Yes. So life is everywhere on our planet. We have life on different environments, extreme environments, very deep, very dark, very acidic, very like this is a river close to the Cap, the Rio Tinto in Spain, in the middle there. Very cold, very salty. Well, there are a lot of extreme conditions where life is on our planet, also deep in the crust, in the atmosphere, everywhere. Life has really colonized our planet. And since the origin of life, life is mostly microbial. We might think that the trees, the plants, the animals like us dominate the planet, but that's not true. The majority of the diversity is microscopic. In the three domains of life, we saw in the nice movies that Carlos showed that life is divided into three domains. The eukaryotes, that's us, we have cells with a nuclei. The bacteria and archaea are prokaryotes, they have a simpler cell infrastructure and architecture, but they are the champion of using all kinds of energy and matter to live on our planet, and they control actually the biochemical cycles of our planet. So life is mostly microbial since a very, very long time, and we are interested to know how it originated on our planet and how it evolved, what kind of traces we can find that allow us to retrace this long history. So the nice picture I just showed, this beautiful life, it's the top of the tree over there, it's life today, and these forms of life have a common ancestor called Luca. We heard about Luca in the movie. So the last universal common ancestor is the most recent ancestor of the three domains of life. It's not the first cell that appeared on our planet. It was quite a complex population of cells with a complex genetic code and a lipidic membrane and so on. So before Luca, there were other population of cells, other lineages that appeared, and that could diversify before Luca. And before these first cells, we have the step of the prebiotic chemistry from simple molecules to the first cell. So when we look at the fossil record, the fossil traces of life, we can try to reconstruct some of these steps, some of these lines that you see there. We can also document forms of life that have completely disappeared, and that for which you don't have any information in the genetic code of modern life. Biologists do this kind of trees by comparing the genetic code of different kinds of cells, endemic trees, with branches that are close to each other in evolution or not. And paleontology or geology can document some forms of life next to these branches that have disappeared. So these are nice complementary approaches. As Carlos said, we can approach the question of the origin and the early evolution of life from different ways. We can use the bottom-up approach by using astrochemistry, chemistry, and some kind of biology to understand how planet forms, or you form a habitable planet or moon where life could possibly originate, even if it's not there. We can also use the top-down approach, looking at the modern biology, the biology of fossil cells, the geology that can tell us about the evolution of a planet, and our planet has changed a lot since it formed 4.568 billion years ago. And the geochemistry, the chemical analysis of the rocks, can tell us how old the rocks are and what kind of environment are preserved, where we can find these early traces. And then the philosophers and the historians of science are really helpful also, because they can make us think about what kind of biases we have when we study all these scientific questions. So when could life have started on Earth? We don't know exactly when and how it started. We can only propose scenarios and test them in laboratories, or test them by exploring perhaps Mars or other planets. And we can have some constraint by, thanks to the astronomers and the geologists. So we know the age of the Earth. The Earth formed about 4.6 billion years ago by accretion of planetesimals. So Earth was really hot and not habitable. The surface was covered by a magma ocean, so melted minerals. So Earth was not habitable, and then cooled down relatively rapidly, differentiated into different layers with a crust, a mantle, a core. And we know that Earth probably became habitable about 4.3 or 4.4 billion years ago. How do we know that? We don't have any rocks from that time, so we don't have any rock record. The rock record starts only 4 billion years ago, so we are missing a big chunk of the first half billion years of Earth's history. But we have something, some pieces of Earth that are tiny minerals. You see down there, the blue mineral, it's called the zircons. And these zircons are very tough minerals, so they can be preserved and recycled and preserved in younger rocks. That's where we find them. We can date them, we can analyze them, and we know from that there was some water on Earth, there was some crust. So if there was some liquid water on Earth, there was some atmosphere, so Earth was habitable. That doesn't mean life was there, but it's possible that life appeared from that time. To find traces of life, we need rocks, because rocks are samples where we can find these traces, and the rock records start only, so as I said before, 4 billion years ago. And I will show you some example of life traces that nobody discusses, because there is a lot of debate in that area. The life that we are sure that it's there on Earth is about 3.4 billion years old. Before that, we have some claim of evidence of life, but as Carlos said, for life beyond Earth, also for life on Earth, we need extraordinary evidence to prove that this is life. You see that also there is a picture of the moon over there, that the moon is covered by craters, and this documents heavy bombardment of meteorites that happened through our solar system history, but a lot of them happened early in the solar system history. So this might have affected or not life if it has appeared before 4 billion years. So you see we have constraints that are quite broad. From 4.3, so not before 4.3, but before 3.4 billion years ago, life appeared on our planet. So there are many scenarios for the origin of life, and Carlos talked about them a little bit. This is a nice one that's linked to the 1 meter pond theory of Darwin, where you have some chemistry happening in the early atmosphere of Earth. There is no oxygen and no ozone layer in the atmosphere yet. You have some CO2, some methane, some nitrogen, and you have lightning, meteorites, the UV radiation of the sun. You can form some simple molecules that will fall in this 1 little pond. That's a hot spring. So there you have phosphates from the volcanic rocks. On Earth, Earth was mostly covered by oceans, but you could have some volcanic islands sticking out of the water where you can have these hot springs. And then in these pools, you have molecules that start to form and become more complex. This is chemistry, this is not life yet. But many people do experiments in the lab and show how simple lipids can form and auto-assemble into vesicles with a double layer of lipids that remind us of protocells. Small bases that you find in the genetic code, sugars, all kinds of interesting molecules called prebiotic molecules that at some point evolve into the first forms of life. This is a gradual process, so it's difficult to put the limit on what is life, what is not. And this is a big challenge when we try to find early traces of life in the rock record on Earth, but also on other planets. Like Carlos said, defining what is a biomarker, what is not, is really difficult. You have all these molecules that form abiotically. You find them in many different places. In meteorites, you can find 70 different amino acids, for example. This is not life. Carlos gave a lot of some examples of that. But at some point, it became life. And we have to put a cursor, and this is a debate. So, big question, what is life? We will not solve it today. Many people talk about that. It's also a philosopher question. We can, nevertheless, try to understand, to define some features that define life as we know it, life based on organic chemistry, carbon linked to hydrogen. Once life is there, it has properties that we can identify. For example, life is made, the cells are made of compartments. These are these lipidic membranes. They have different composition, but they are lipidic membranes. Life reproduces, and life has different kinds of metabolism to take out energy and matter from the environment or produce it, to grow, to maintain itself, to divide, to reproduce, and so on. And another characteristic of life is evolution by natural selection. So these characteristics are important feature, and we can try to find evidence of that in the geological record to prove that what we observe is really life traces and not some produce of abiotic processes. So what do we do when we look for early traces of life? We cannot look for big dinosaurs or some DNA, of course. This is not preserved. I'm talking about billion years old rock, not early fossil, not young fossil, like some million years old fossils. And we will use different kinds of tools, going from the macroscopic scale, going in the field to get these sample of rocks. And then we can perform all kinds of analysis, like we do in biology. These are biological traces, only they are fossils, and they've been modified by different processes. From the macro scale to the nanoscale, and even using tools attached to a synchrotron to have really high resolution observation of these possible traces of life. We can look for 4 kinds of traces. The two on the top, there are morphological traces of life, either rocks produced by life. You have microbes that can precipitate or trap sediment grains, rock grains if you want, and construct big structure like stromatolites. You see this here is just one big stromatolite. That's 2.5 billion years old, and you see the people just before. And we can also look in modern environments what kind of traces life produces, like this picture above on the left side over there. These are stromatolites that are growing right now in Shark Bay in Australia. So we can study them and then compare them to the oldest record and try to understand how they form. Of course, they are not the same, but studying modern life and see how this can be preserved help us to detect and interpret very old traces. Over there on the right we have fossil cells that are preserved in different ways. They can be preserved just flattened in mud, and this mud become a rock and you can extract them from the rock. Or they can be replaced by minerals, covered by minerals. All of this depend on the environment where we are. The two examples at the bottom are chemical traces of life. These include isotopic signature of elements that life uses in its metabolism, like carbon, iron, nitrogen, sulfur, and so on, or metals that are in the heart of enzymes that life will use and that can be preserved within fossils. We can also look, of course, for fossil molecules, these biomarkers that are present in the membranes of organisms, in the cells, structural lipids, or so on, and so these are the kind of tools of traces we are looking for. So this is challenging because as we go deeper into the rock record, because we live on a very active planet, it's made of, the surface is made of a crust that includes different plates, and these plates move, they create oceans, they collide and create mountains, there are volcanic activities. Rivers can erase a full continent, they can just erode mountains. You have the wind or ice that can also erode rocks. So all of these geological processes can erase the rock record and erase the fossil traces of life that could be preserved, perhaps. So this is one of the challenges of this adventure. Another big challenge is the fact that some abiotic processes, chemistry or mineral formation, can mimic life. They produce forms like the one you see on the bottom right. You see these little spheres, or this more complex sphere, or you see these filaments that can be curved or not. All this is made by abiotic processes. And this can resemble fossil cells or fossil filaments that you can find in real life. So at some point it's difficult to discriminate between biology and abiotic processes, and this is even more difficult on Mars or beyond Earth anyway. But we have to develop criteria to distinguish both, and the context, the environmental context, is very important to discriminate them. So here are some examples of the oldest traces of life on which everybody agrees. There are proposed older traces before that, but they are very debated because there are abiotic explanations that can also explain what we see. So we are in the fuzzy area. Is it life? Is it not life? It's still interesting to document, but we're not sure. So the oldest traces are at least 3.4 billion years old. They are these stromatolites. Before I show you stromatolites that has higher than this room, but the first ones, they are very tiny. They are a few tens of centimeters high. But they combine all kinds of information, not only their morphology, but their structure. They preserve organic matter that has an isotopic signature of metabolism made by bacteria, for example. They have a shape that suggests photosynthesis, bacteria using sunlight for its metabolism. So all the features combined suggest strongly that this is life. So when we study all traces of life, we don't use only one criteria to say this is life. We really need a set of criteria and understand how it was formed, how it is preserved, how it is modified by geological processes. We also have fossil cells that are 3.2 billion years old, microbial mats in marine environment, but also in terrestrial environment, in fluviatile environment. So by that time, life was already diversified in different kinds of environment. And we have different metabolism, different evidence of cells using light producing methane, using sulfur, iron, nitrogen, carbon. So really a diversity of metabolism that we know in modern bacteria. That means that LUCA is older and the origin of life is much older. Oops. Can I go back one? Thank you. So I make a big jump in time. Another big step in the evolution of life on Earth is the rise of cyanobacteria. So cyanobacteria are a kind of bacteria that use light for their metabolism, but they produce oxygen as a waste. They split water and they reject oxygen. And this has a really deep effect on our planet. That's thanks to these bacteria that oxygen started to accumulate in the ocean and the atmosphere. And then now we have a planet with a lot of oxygen. This was progressive, and this started more than 2.4 billion years ago, but became permanent 2.4 billion years ago and then increasing with time. So these cyanobacteria are very important in past and modern ecosystems. They're also important because they are the ancestor of the chloroplasts that we found in algae and plants. They produce oxygen that many eukaryotes need to make their lipids or to live. Some don't, but many do, and so the rise of complex life is also linked to this increase of oxygen. You see examples here of fossil cyanobacteria that some of my PhD students study and document, so they can be really preserved, as you see there. These are macrofossils. Then another big step, the first, the earliest fossil cells that document the evolution of eukaryotes or domain are at least 1.75 billion years old. They are preserved in mud, so mud that became a rock, and we can extract them and do all kinds of analysis to prove that these are eukaryotes. They have complex appendages, they can have a whole microbial place, they have all kinds of chemistry also that tell us that they are eukaryotes. Another jump, 1 billion years ago, we see evidence of a diversification of algae, green algae, red algae. These are multicellular algae, that means they have a unicellular ancestor that's older than that. So we start to have a more complex ecosystem. We also have fungi that appear by that time. This is important because fungi are part of a group that includes also animals, us. That means our common ancestor is older than 1 billion years old. Another jump, around 558 million years ago, the oldest microfossil of animals are preserved. See that they are a bit different from the animals we know today. The one you see in the middle or on bottom top over there is called Dickinsonia. It's an organism that can be up to 1 meter and 40 centimeters long, so it's quite big. But it was moving on the seafloor and then just dropping on microbial mats and absorbing organic matter and then moving like that. So we have a modern analog over there. You see this little, this is microscopic, it's not big, but it feeds the same way. So we have a diversification of animal life at that time, forms that have completely disappeared, but also ancestors of life that we know today. So when we combine all these elements, I'll just show you a few examples, but when you combine all this information from the geological context, the evolution of Earth, going from an Earth without oxygen in the atmosphere, with not much continent, just a few volcanic islands, to Earth with different kinds of continents. Sometimes they collide and make a big continent, sometimes these fragment, and this happens at least 5 times through time. It will happen again. We have also big glaciations, we have meteoritic impact, big volcanic events, all kinds of geological processes that affect the evolution of life. But we also see that life affects deeply the evolution of our planet with, for example, or especially these cyanobacteria that change completely the chemistry of our planet and that allow the evolution of more complex life, including us. All this is linked to chance, perhaps a little bit of a constraint, but a lot of chance. So if we erase everything, we start over, we will not have the same thing. So it's sure that if we look for life beyond Earth, we will not have the same thing. So our planet is a biological planet. When we see that, we see plenty of evidence of life. Of course, we see the technologically advanced life over there, but we can also see in our atmosphere that there is life on our planet, in the rocks that are preserved, in the water. Lots of traces of life over there. So we can wonder if we are alone in the universe, because life is known on Earth only right now, but we didn't explore a lot yet. We didn't have the tools to look for life beyond Earth until now. Lots of people thought that there is life, plenty of life everywhere, since the ancient Greek time, that asked all the right questions and thought about everything already. But we are lucky because we have now the technology to really look for life beyond Earth. So all this knowledge of life that we have on Earth allowed to develop this concept of habitability, which is the possibility for a planet or exoplanet or moon for life to begin, which is not the same as for life to persist. These might need other conditions. And depending on there's a lot of literature on this, and depending on what you prefer, the scenario you prefer, you think we at least need liquid water, some chemical elements. Liquid water can be liquid if you have a thick atmosphere or if you have an icy crust. We need gradients, thermal gradients, chemical gradients, some UV light, some lightning, a stable climate, oddly, to have perhaps the origin of life on a planet. So this concept of habitability is interesting, and it was developed by astronomers to understand more or less which planet could be habitable depending on where it is compared to its star. But habitability is also a concept that can be changing in time and space. For example, on the bottom there you see on the left side Mars. Early Mars was habitable. There is plenty of evidence of liquid water, running rivers, oceans, and so on. But early Earth, very early Earth, was not habitable, then became habitable. Above there you see Europa, that might be habitable or not, where we have water, probably energy and elements, but maybe it's not enough. It's also a debate. And then we have all these exoplanets, including TRAPPIST-1 that was found by my colleague in Liège, Michael Gillon. TRAPPIST is a beer, but it's also an acronym. And the next project he has is called Speculoos. It's also an acronym, and it's also valid. And with James Webb, we will have a lot of more information. So right now we are looking for life on Mars. This is a way to test our hypothesis. What are the conditions necessary for the origin of life on the planet? Did life appear on Mars, or do we have some kind of complex prebiotic chemistry? Where did it stop, and why did it stop? So we can compare the early history of Mars with the early history of Earth. And we see that the beginning is similar. Once they cooled down, they were habitable both, but then quickly Mars became inhabitable at the surface. It lost its atmosphere, its magnetic field. Earth stayed habitable, and we have the earliest traces of life 3.4 billion years ago. A great thing on Mars is that the oldest rock are preserved. I told you on Earth the rock record started 4 billion years ago, so we are missing half a billion years. And this half billion years is preserved on Mars because Mars is a less active planet. It's active, there is volcanism, but there is no plate tectonics, so there is no plate moving on the surface. That means that the oldest rock record is preserved. And this is really interesting. Whether or not we find life, it's interesting to study these rocks and see what kind of conditions happen there. It's close to us, so it could tell us something about the conditions of the early Earth that we lost. So there are several missions exploring Mars. ExoMars, we are part also of ExoMars, a mission by ESA. It was postponed due to the war in Ukraine. It was supposed to leave in September, and now it's postponed to 2028. That means we have more time to define what is a biosignature, what is not. We are doing artificial fossilization of life forms and see in Martian conditions and see if ExoMars can detect it, for example, and so on. So this is my last slide. So astrobiology, it's about life here and life there, perhaps. It's about the study of the origin, evolution and distribution of life in the universe. And thank you.
Thank you both for these very inspiring talks. And now I would like to invite both of you at the same time to be on stage. And now I also invite the public, if you have some thoughts that have popped up during these talks, some questions or just speculations that you may have, they'll be happy to discuss with you. We have already one. Maybe. Is it working?
Okay. Thank you very much for your presentations. Were like very interesting, really. So I have a question for both. You are trying to answer the question of a philosophical question, actually, and also to understand better our past, the past of Earth. But I want to ask you, like, do you think if someday we'll know about this question, we know the answer, will this help us in the future? And like, can you guess in what will help us as a, like, you know, humanity, like, just to know more about us, about how we, like, I mean, it's a very strange question, but
well, it's a very difficult first question, of course. It's a philosophical and epistemological question. And is this working, by the way? Yeah. Okay. I think that the kind of questions we think about are I mean to develop our brain. I mean, if you are only thinking about how these nice pieces were constructed, your brain becomes perhaps less and less complex. Okay, but if you try to answer how life originated, how, for instance, the chemistry became biology, how complex organisms start moving around, and how our brain itself developed, your brain as a human is increasing. So science is, in my view, science is a key. How to explain in short words? Science is essential to behave as humans, in the sense that you have to face very, very complex questions. The development of science along the history has allowed us to construct more and more complex, for instance, instrumentation, that in turn has also stimulated new questions and so on. So if we learn something about our origins, our cosmic and terrestrial origins, we will be able to be more humans, and then to be able to develop technology, nanotechnology, microbial-based technology, biotechnology, whatever, but which is inspired probably in those questions that can help you in the future, and also in a practical way. So this is my short try to answer to this very complex question.
Like Carlos said a lot already. I think we are really curious, so we want to know, and I don't know if we will ever know how it happened, because we will not find any evidence of the origin of life, except the fact that we are here, but how it happened. We can just say this scenario is more plausible than the other. But I think it would be really nice to understand a plausible scenario and to find traces of life beyond Earth. We are only one species, and we are only temporarily on this planet. We will disappear. We are not eternal, but other forms of life will come after us on Earth, and we are accelerating the process actually right now. But microbes, they don't care. They will be there until the sun blows out. And it would be nice to know if there are microbial life beyond Earth. We look mainly for microbial life because the origin of life, from what we understand now of the conditions, it seems not so difficult considering the number of planets and so on. So it would be nice to know that there is a lot of life, plenty of different kinds of life, and we are curious to see how they look like, what they do, how do they live. Complex life, I don't know if there is complex life beyond Earth, perhaps, but complex life beyond Earth following a lot of chance, so life elsewhere will not follow the same path, but it doesn't matter, but just we have an end, our planet is the end, our solar system is the end. It would be nice to know, to understand the origin of life beyond Earth and if there is life somewhere else. But just as a natural process. Then other people might have a big impact on their faith if they are religious. This is an impact on society, and most humans believe in something. That's another question. So I think for different questions, for scientific curiosity, for just our place in the universe, we are just a tiny planet in a tiny side of the tiny galaxy. But and even on the tree of life, we are just a tiny point on a branch. We have a big impact now, but still we are just one form of life. It's just, maybe it helps to appreciate also that life is precious on Earth, whether there is life beyond Earth or not. But sorry for the answer, it's just...
Perfect. This is what we want. We have another question over there.
Good evening. What do you think about the possibility of exotic biochemistries? Do you think it's worth it, for example, to devote resources to look for life in Titan, for example, when there are candidates in our solar system for a normal carbon-based life?
A lot of people ask this question about life based on silicium or something else, or not in water. But the thing is that everywhere where you look, you have organic chemistry. Organic chemistry is not biology, and the chemistry of carbon linked to hydrogen. It's really abundant, banal in the universe. You have a lot of water too, and liquid water, especially liquid water, has very special properties that no other liquid has. And the chemistry of organic chemistry in water is very rich. It can make a lot of different things. Organic chemistry can make very complex molecules with complex information. It can make all kinds of things. When we talk about looking for life beyond Earth, it's not the same life, of course. It's some kind of life based on organic chemistry with liquid water and minerals. If you look beyond Earth on what kind of chemistry silicium makes, for example, silicium makes minerals beyond Earth also, not life. Even though silicium can link to 4 atoms like carbon, the links are stronger, so you need more energy to break them. You have a less complex chemistry, a less rich chemistry than with carbon. When you look in meteorite, you find amino acids. You can reproduce easily with the Stanley-Miller experiment amino acids, bases, sugar, the bricks of life, even if it's not. So there is a lot of characteristics of this organic chemistry that point to, seems that if there is life elsewhere, it will be based on organic chemistry. No, that doesn't mean that it will not be, it will be exotic by comparison to Earth. You can have, for example, we are working on the possibility of life on TRAPPIST planets. These planets, they turn around a star that's very tiny and that's sending light mostly in the infrared range. So that means possible life on one of these planets will use another kind of light. These planets, they are locked to the star, so they always show the same side. So there is one side that has light and one side that is always dark and cold. So these are very extreme conditions. So if we find ever evidence of life in the atmosphere of this planet, that would be very special and different. we have actually on Earth we have some form of life that use infrared light, so we know it works on Earth. So try to understand if it's possible to have that kind of biosphere on these TRAPPIST planets. So in Titan, there is a lot of debate about Titan. Titan is very cold, so the liquid is methane, and at this cold temperature, any chemical reaction is very slow, so it doesn't seem very probable that there is some kind of life there. But you know, some people have other opinion.
Also in Titan, in carbon-based liquids, the problem is that they are not polar. So the polarity is changed, and the membranes, you should imagine a different kind of membrane with a non-polar side outside, and the polar heads inside. So it's very difficult to imagine, but of course I agree with Emmanuel that as far as we know, all lives, all examples of life we can find in the universe should be, let's say, based on carbon and water, because from a chemical point of view are the easiest or more open way to produce complex biochemistry. By coming back to your original question, the biochemistry itself could be very different, even based on carbon and water. For instance, you can imagine different polymers to store genetic information, apart from DNA and RNA. Those have been very successful here on Earth. Those were perhaps first RNA, and then DNA was present in the very early history of our planet, and the origin of life was probably based on RNA. But if you can ask a student of organic chemistry, for instance, about the possibility of constructing a polymer to store genetic information of a kind of heritable information, perhaps the student wouldn't select a polymer made of phosphorus, phosphate group, and ribose, phosphate ribose, and some nucleobases, and then complex helical structures. This has been the solution here on Earth, but perhaps life on other planets or satellites, even based on carbon, has selected somehow a polymer very different, so different that perhaps we cannot imagine how different it is from DNA and RNA, so nucleic acids. There are some groups, for instance, here in Europe, working with some molecules that mimic natural nucleic acids, and with some properties which are similar and others are different, that have they have a technological they were originated facing technological problems, biochemistry, biosensors, and so on. But apart from this technological interest, they can give you possible solutions for the store of genetic information in other places. So the biochemistry could be very different. When you see the metabolic chart of a cell on Earth, there are lots of metabolic options that could be very different, and also could be in the different enzymes or enzyme-like molecules performing metabolic actions could be very different in this map. So we have to be very open to different biochemistries and metabolisms in different parts of the universe, of course.
Yes, I would like to ask about this question that you raised as a way to interpret the origin of life, about whether it was by chance or by necessity. I find it a very interesting angle or point of departure for these kind of questions. And in particular, I wanted to ask your views about the drivers. If it was by necessity, what would be driving that necessity? Some kind of competition, selection? Why? Because I mean, everything about life is going towards more complexity. So it's very counterintuitive, isn't it? When you will expect the systems to evolve to minimum energy, minimum complexity, etc. So what is driving life?
It's also a very easy question, okay? And we don't have beer still, okay? Chance and necessity is the two options, the extremes of the gray scale, no? For the origin of life. You have, for instance, scientists like Jacques Monod, who was in favor of chance, pure chance, he said, or Christian de Duve, from here, from Belgium, who was in favor of a kind of deterministic origin of life once the complexity of the mixture of molecules was enough. So what drives necessity is complexity, let's say. A complex system can develop processes and jumps in complexity that are more than the sum of the parts of the system by definition of complexity. And perhaps once you have a collection of organic molecules with that are interacting among them and constructing such a complex scenario, life starts. We don't know how. We are investigating on the possibilities of changing, for instance, metabolic molecules, so proteins or peptides, mixing them with vesicles, as Emmanuel said, and also nucleobases that can form nucleotides that can form polymers and so on, to mix everything together and see if something happens. That could be a kind of a procedure that can tell you that the complexity has to reach a certain level that triggers the emergence of a new property, in this case life. But chance is everywhere, and chance in this planet is of course changing the behavior of all chemical and biological systems from the very beginning. And perhaps I would say, and I think that most of us would say, that a mixture of an initial increase of complexity, so necessity in that sense, mixed with chance, with some events that were there and could have not been there at that time, were tracing the origins of life. All the branching process that Emmanuel showed, and at the end, different chances, different possibilities were moving the shape of the tree towards one side or another. So I would say that it's a combination of chance and necessity, and the point is that even if we were able to understand this combination of chance and necessity in the origin of life here on Earth, would that combination be the same on other planets, for instance? We don't know. So, but the question is very, very complex. Yeah.
I'd just like to add that evolution doesn't go always toward more complexity. There is a lot of simplifications also, or not much chance sometimes, at least in morphology. The genetic code continue to change, and evolution doesn't stop. But if we look at the tree of life now, we are there, and all the bacteria are at the same level as us, and the archaea, everything has evolved. There's just some different kinds of structure or metabolism. The bacteria that live today, they have a long, 4 billion years of maybe less, of evolution too. So we have to be careful not to think that we are some kind of top of the evolution and stuff, and that there is no direction in evolution either. There's a lot of chance, like Carlos said, once some molecules meet each other, they will form. You can predict what kind of chemistry they will form in different pH, temperature, and so on. And this condition of pH, temperature, these little pools on early Earth, they exist by chance because of some geological processes. And then by chance, molecules form and they meet, and then they form. They have to form such molecules that are a bit more complex because this is chemistry. And if you have some catalyst, like a phosphate, this accelerates that reaction, and that means this molecule are more abundant than the other, and so on, things like that. And then at some point we have life. That's a question.
Keep it.
Who has a simple question?
We have 2 minutes for
Maybe it's simple.
No, but it's great. It's always great to discuss about these things.
Regarding the biomarkers, particularly very interesting because I work in biomarkers in other field, more oncology. Because mostly of these, so basically we didn't find still any biomarkers, no. Could be that because they're not stable molecules. Because their variable of time in different plants we don't take into account. Stability of that molecules we don't have tools still to analyze how they compound. Because mostly of the chemical reactions, like methane, they are reactive, but they are more stable because they keep in the reactions. But mostly biological biomarkers, they're actually not stable.
Well, some of them are stable enough that you can find them fossilized. For example, the oil that we put in our car, it's made of biological molecules. It's made of lipids, of pigments, and we can find them. As the oldest one we found is 1.700 billion years old. The limitation there is the preservation, because once the temperature that the rocks have seen is too high, then the molecules disappear. So that's one limitation, but that's not the only kind of molecule that can be preserved. I showed you some fossil cells that are 3.2 billion years old, and these are organic walls, so the molecule that made them are preserved, but they have been modified. So this is difficult to identify who made them, but these are biological in origin. So we can find old biomarkers, but at some point it's difficult to know what can be produced abiotically, what can be produced biotically. For example, DNA, RNA, you can form them in the lab, but naturally they will not form. But they are not very stable. But some pigments can be maybe more stable and preserved for billions of years. So we can imagine on another planet, if life needs pigment to use the light or to protect itself as a sunscreen, like life do on Earth, perhaps these molecules could be preserved. They will not be the same as on Earth, but perhaps complex enough that we don't understand how they could form abiotically. But these are not, so when we look for traces of life, we don't use only molecules. We really need a combination that's not only molecule, but also morphologies, structures, things that cannot be explained only by physics and chemistry that we understand now. So.
Yeah, in fact, it's different, the kind of biomarkers you can expect in extant life and an extinct life. Yeah? So, for instance, when we go to Mars, perhaps we can find some combinations of molecules telling you that life was there 3 billion years ago. Okay, and these molecules should be lipids or some molecules similar to those shown by Emmanuel at the beginning of her talk. But the combination of them and the geological context and so on has to be taken carefully, because it's a combination of issues which can tell you that life was there or life is there. And that's why in some of my take-home messages, when you read that, we have found this molecule, so life is there or was there. No, it's not so easy. And of course with small molecules, and of course with those molecules that are elsewhere in the universe. So it's very difficult to find a real biomarker or biosignature, and to be able to convince the community that you have found something that is telling you that life was there, in the case of Earth, and life was there on Mars, or life is there, for instance, in Europe or in Enceladus. So that's why we have to be patient.
So life has evolved in a way that makes us feel hungry when we need to eat. So
well, this is metabolism. It's one of the 3 components of life. Yes.
So you can continue asking them questions, but at 9 p.m. we have to finish all the food that we have prepared for you. So I would like to thank again the 2 speakers.